thing to think about is that the phosphorylation then is always coupled with the dephosphorylation. So you can, every reaction is reversible. Okay, that's also important. Every reaction in the signaling pathway is reversible. Okay, if the protein kinase is less active than the protein phosphatase, then the protein's unphosphorylated. So the phosphates are constantly being removed, unless you want to inhibit the phosphoprotein phosphatase, which sometimes you can. Okay. Here's the part of in the book that I disagree with, and actually the book disagrees with its own figure too, but so I don't know why they even left that figure in there. So in the book shows you that the phosphorylated form of the protein is active, and the unphosphorylated form is inactive. You've already seen an exception to this rule.
EIF2.
EIF2, exactly. That's what I was thinking about too. So when you phosphorylate EIF2, that's inactive. And the book concedes, the book does concede that, well, sometimes the phosphorylated form is inactive. So why did they leave that figure the way it is? Okay? So that's figure 15.7. So just ignore the little activation part. Because we are also going to see sometimes that phosphate group is going to mark the cell for ubiquitination. So that's really not good. Okay, so while phosphorylations usually lead to activation, it's not a general rule. Just sort of ignore that, and make sure you understand what the phosphorylation event is going to do to the protein. Okay, so that's one. We're going to encounter gobs of protein kinases. Okay, here's another one. We've already talked about them. Okay, GTPases. And there's lots of different types. So generally these are called G proteins. Okay, so a G protein coupled receptor suggests that we're going to involve a GTPase. And this is okay to say that a G protein requires a GTP to be active. Okay, so we've hit lots of these so far. We've hit ARF and SAR and all of the RABs. We've seen EIF2, the signal recognition particle and the signal recognition particle receptor. So we've already encountered a lot of G proteins. If I do this, that means the protein is active. It can do whatever it is supposed to do. Okay, so we know that there's going to have to be a guanine exchange factor that will pull off the GTP and replace it with a fresh GTP. Okay, and we've seen a couple of guanine exchange factors. You did those really well. SEC12, SEC12 was one, and EIF2B was one, and you did fine on the test with those. Okay, immediately our first guanine exchange factor that we're going to encounter are G protein coupled receptors. And you also know that even though the G protein is the GTPase, and I didn't see anybody get that mixed up on the test, so I was very thankful for that. Sometimes you have to punch the protein and the activated GTPase activity, and that's what the gap is for, GTPase activating protein. Okay? A lot of times it's the effector, that the thing that the G protein binds to happens to be the gap. Some people will say that the G protein used by G protein coupled receptors has its own inherent gap, and that is true, but you can just kind of accelerate the hydrolysis of GTP with other proteins. Okay, so you're going to see lots of G proteins. Not just the proteins that are activated by the G protein coupled receptors. There's lots of those. You're going to see probably the other big one is RAS. This is the one probably the most significant G protein we'll encounter. Okay, so do you guys like your test? You know what? I did not calculate the average, and I'm really sorry.